So this huge essay is my last slide. But let's just say, this all has to do with Goldschmidt and the Beaver's talk, and it's the mechanism of purifying selection, telling you the truth. Regulatory complexity could increase. It does increase, because you get more regulatory genes and any other connected gene. You get more transcription factors. And as repeated tetraploidies occur, until they're mitigated, they could be mitigated by such as a chaperone. Anything that keeps the complex from feeling the concentration of any component. If it's guided in, then it won't feel it anymore, okay? But apparently that's not the case, because they keep building up. If that's the limiting step to regulatory complexity, then this drive, this repeated tetraploidies, could explain why plants have definitely gotten morphologically more complicated, maximally. From 450 million years ago, that's when we had the first land plants, the liverwort kinds of plants, the Cooksonia fossils, those little sporangia-like things, to what we call a plant today. There's no question there's been a stepwise increase in morphological complexity, not complexity in general, just morphological. And this could explain that, and it explains it purely neutrally. Tetraploidies, they just happen. Purifying selection, it just happens. Biased fractionation, it just happens. But as a spandrel to all that stuff that just happens, connected genes build up, pushing evolution in a direction. Goldschmidt would love that. Everything's neutral, everything just happens. But as a spandrel to it, and that's the correct word, as a spandrel of purifying selection, morphological complexity could be pushed in a direction going up. And that would be Goldschmidtian. Thank you. I'm only 5 minutes over. I was very worried I'd be hung up on the past. It's easy to be hung up on the past.
So, are there any questions?
Yes?
So I'm not sure if I understood it exactly, but what do you think is the mechanism or the reason for the bias fractionation?
Yes. First, both in Rhodopsis and in maize, every region you look at is biased to one or the other, but it doesn't absolutely mean that there's one genome going into the allo. It's just a parsimonious hypothesis to explain how it's so even covered, the bias is evenly distributed over the whole genome.
Is it just big bytes, Mike?
No, it's not big bytes, it's number of bytes.
Okay.
It's not the length of the bytes, because we looked at the over and under stranded, we got the perfect same distribution of byte sizes. So it's no, it's not. That's a great question though. It could have been length of byte, which would have been very cool, but it's not the case. The way I look at it, 2 genomes in the same nucleus, one is simply thought of in terms of small RNA biology as an invader, and it's marked, so that it can't move right recombinationally, and therefore it can't delete right. It's marked for attack by some way, and as a byproduct of marking it for attack, it's purely epigenetic. I can't think of anything but some evidence, and we are working very hard to find that tag. It's certainly not raw methylation in any of the formats that we study with bisulfite sequencing. We know regions, we've sequenced enough bisulfite sequencing to know that that's not going to give us our clue. But we haven't looked at the histone tags yet.
So you're creating tetraploids and
No,
watching this?
we know in maize that the bytes taken today, the bytes that are different just in the lines of maize, go to the over-fractionated chromosome. We can predict where the genes are being lost. Today, that tag is still there.
Still going.
Yes. So at least we know that the tag's still there, and so we have a logic to look for it. But we haven't found it. What would you look for? I tell my lab, that's where Z-DNA could come in, or something equally really mysterious, to make it so that the supercoiled chromosome is somehow stiff. So it can't make those loops. So it can't do the recombination event. Or similarly, that it recombines outside of the window of recombination. So its entire cell cycle time is lagged, or something that's equivalent. It's just all just guesswork. I think it's because the epigenetics itself is just hand-waving. We don't have a single piece of evidence for epigenetics. It's a default hypothesis. But I think you would agree that it's worth going after, because if you nail it, it would be a nice thing. Yes, we don't know why bias, but in the two places where it's been studied intensely, it turns out to be the whole genome, both in Arabidopsis and in maize, the whole genome is biased and fractured.
Have you looked at evolution of the plastids relative to these changes? So are plastids in different lineages showing patterns that are... Plastids between monocots and dicots in grasses are practically, they don't have any gross abnormalities. But if you take and look for teeny little movements, so here's a plastid genome, chloroplast genome. A little piece of DNA can move from here to here, and that can be an excellent marker, and is generally not counted by those who look for, people are often trying to use the sequence of plastid chains to make their trees. They'd be way better off to look at the breakpoint associated movements of small pieces of DNA in plastids from here to there. Very unlikely to revert. Very likely to be a character, an irrevertible character of it in its own right. Between monocots and dicots, there's one large inversion, and otherwise, collinearity goes all the way back in a plastid to green algae. And you can't see a thing at the nuclear level. So the level of conservation of genes in gene order is way greater in a plastid than in a nucleus. So it's great for deep roots. Better for deep, and the nucleus is way better for species and ecotypes. Yeah.
So I actually didn't understand a little bit about the tandem repeats.
Yes.
Are they, how long are they? Is this just random
yes.
DNA that's dispersed?
That's right. So tandem repeats come in the classical category, whole genes being repeated, and 10% of the genome is those kind of things, big repeats. And then we just look at the DNA in a plant, and it's just full of tandem repeats, even in Arabidopsis, where it doesn't have many transposons. But if the average resident between your genes is a transposon, as is the case in maize or humans, they are loaded with repeat DNA, so there's enough transposons or there are corpses left. They just leave repeats, and every one of them has some chance to do this, and it's all just a big bag of potential. And you can look down there and you can make thousands of little loops. And among those, some of them, for reasons unknown, are perhaps better than others and actually create a deletion from 10 to a kb long. And that's a byte.
And do you have a sense for what molecular pathway is actually doing this?
The only pathway I know is regular mapping reciprocal recombination. Because the arm lengths in map units for over and under biased fractionated in maize don't go together. They absolutely don't care whether it's the over or the under, so it's not reciprocal recombination of the map unit variety. So it's still reciprocal recombination, I think, but I think it might be called, there's a word, not ectopic, weird, no. Offbeat, not regularly studied, not rad2 maybe. I don't know, and I have, my colleague Zach Candy is going to try to count little recombination mechanism markers that can give you the rate of recombination in different chromosomes and see if you can get anything that respects the over-under fractionation in maize. And they have big chromosomes. So maybe RAD2, for example, little RAD2 immunospots on the chromosomes, count those up. Maybe we can get a 2.3 to 1 ratio, and that would be very cool for something like that. But the answer is no. I don't know anything about mechanism. And that's why I like my new line of work.